So einen schönen Abend und willkommen zum Kolloquium der Plattform Biodiversität. I will switch to English, of course, because today we will have an English-speaking guest. I'm happy to present Heidi Haufe, and despite her German-sounding name, she's Canadian, but she has been working in Italy for quite some time now, and since 2008 she is working at the Fondazione Edmund Mach in San Michele in Trentino. And since 2011, she's actually, and I have to read that, the leader of the Conservation Genetics Unit and head of the Biodiversity and Molecular Ecology Department. So I'm quite happy that you are here today. And I know that your research focus is actually on conservation genetics and meta-taxonomics from a very wide range of organisms. And I think we can see that already from the title, which also includes monkeys and microbiota and meadows. So thank you, Heidi, for coming, and I give the floor.
Thank you. Thank you, everybody. It's really nice to see such a big audience this evening on such a cold night, so thank you very much for coming. Yes, I am Heidi, and I hope English is okay this evening because I find it easier to explain my science in English. So right away, here at the bottom of the screen, I wanted to mention right away my main collaborators, the main institutes that have produced this data with me. So Cardiff University, the University of Florence, EURAC Research here in Bolzano, and the University of Innsbruck. As Julia said, I am at the Fondazione Edmund Mach, and just in case some of you don't know where that is, it's at San Michele all'Adige. It used to be called the Istituto Agrario di San Michele, which of course is still there. It's part of the Fondazione. In 2008, 2009, the Center for Alpine Ecology on Monte Bondone and the Istituto Agrario di San Michele were put together, and so we became the Fondazione Edmund Mach, still funded mainly by the province of Trento. The center is made up of three centers, the institute, the Fondazione is made up of three centers now. So the Istituto Agrario is still there, and it's now the Center for Education. The Technology Transfer Center, which is a lot of technicians who go and help the farmers do a better job, and the Research and Innovation Center, where I work. We are now, under the new director, 20 research units and 20 technological platforms. If you want some more information, there is the website at the bottom of the slide. So what is animal biodiversity? Most of you will already think of biodiversity as a number of different kinds of organisms, mainly the ones that you can see, how many live in the same ecosystem. This is species biodiversity. A lot of you will also know about genetic biodiversity, which has only in about the last 20 years become very important for conservation as well, because it's not enough to have a lot of each organism for it to survive. Those organisms in their populations also need to encompass a certain level of genetic variability. That is, they are slightly different between each other so that natural selection can occur. Without variability among individuals, the species does not survive either, because it becomes very prone to getting new pathogens. It becomes prone to inbreeding, which leads to genetic diseases and so on. So genetic diversity is also now a well-known biodiversity. But another biodiversity which has been ignored for a very long time and has only just become a focus of study for many scientists is the microbiodiversity or microbiota. So you can have these are microorganisms or even very, very small organisms. So you can have microbiota in the soil, for example, microbiota in plant roots, microbiota in the tiny soil organisms that live in the soil, and in domestic and wild animals, in our gut, on our skin, and so on. And this microbiodiversity in the last 10 to 15 years has been shown to be extremely important in human health, but it hasn't been studied very much in the wild, as you will see. So why do we study biodiversity? Well, it's been recognized at a national and international level that we can't afford not to think about biodiversity. I think this is very clear to all of you, having all been through COVID, having heard about all the stories of how, because we lack biodiversity, we are getting in touch with wildlife, how this means we are pathogens are passing from them to us. A high level of biodiversity in our natural ecosystems, but even in our urban ecosystems, is very, very important to humans and for life on Earth. So we have the national strategy for biodiversity in Italy. There's the Convention on Biological Diversity. We have all the European directives. We have to save habitats, birds. We have to stop wildlife trade, invasive species. It's well recognized, even at a local level, that for agriculture we need biodiversity, and for our quality of life, and even for tourism and economy. In Trento, at least, tourism, which is mainly based on the beautiful outdoors, is worth 15% of its entire economy. And biodiversity is important on a global level, as I said. Alpine biodiversity is more important than people realize. Even though 20% of the population on Earth lives and 20% of the biodiversity is in alpine areas, the biodiversity and the services that they provide are provided to 50% of the global population. So 20% in the mountainous areas serves 50% of the global population. So it is extremely important. And at an economic local level, we need biodiversity for our tourism, as I said. We need it for the alpine traditions of farming. And we need it in agriculture, for pollination, for diversity in our cultivations, for maintaining our cultivations, for insects, etc. We even need it for our public health. We don't need to repeat it again. Our biodiversity helps protect against invading pathogens and diseases. So, although we often think of biodiversity as a number of beautiful habitats, we like to be out in it, we have many different habitats in the Alps, we have many different species that help to keep the water clean, that help to give us new pharmaceuticals. 50% of pharmaceuticals still come from natural biodiversity. We need to remember that biodiversity is essential for the fertility of our soil, for absorbing the pollution that we produce, for protecting us against invasive species like the tiger mosquito and other new vectors, and also against the pathogens themselves. So just as an introduction to what my unit at the Fondazione Mach does, we look at patterns of biodiversity and genetic health. We use genetics and genomics to look at the health of populations and species so that we can give scientific support to the management at the political level. So we do basic research at an international level. We have 25 years of experience in animal genetics and genomics, mainly wild animal. We are particularly specialized in using non-invasive samples, so we collect fecal samples on the territory, or soil samples, or fur, feathers, etc. We don't need to trap the animals anymore or hurt them in order to do our work. So we can do analyses of environmental DNA, of antique DNA, and we can also do forensic genetics for wildlife. But our main goal is always to end up with concrete applications for conservation. So we have three main objectives. The first is to study the genetics and genomics of alpine species. And that ranges from fish and amphibians and birds and mammals, using mainly DNA, non-invasive DNA. We also develop new genetic and genomic tools that can be used to monitor genetic and genomic biodiversity, and we use metabarcoding for that, so techniques that can take a sample, extract all the DNA, and tell you all the species that are in it. We can do this from water, from fecal pellets, if we want to look at diet of an animal, for example, or also soil. But today, I'm mostly going to be talking about our third objective, which is looking at the role of microbiodiversity in conservation. So ignore these words for a minute, because I'll explain them in a minute. But we are mainly looking at how the gut flora and the skin flora is important to conservation of wild species and ecosystems. Yes, for this we use metataxonomics mainly, which I will explain in a minute. Of course, I don't do this by myself. My group is now has 23 people. There are 8 of us that are permanent, all of us with different specialities. So I have people specialized in birds and mammals, in fish, in carnivores. And then I have people who analyze data, and then people who work in the lab doing the actual lab work. 8 of us permanent, and then I have a number of doctorate students and stage students from various universities. Just so you know, I'm not doing all this by myself. And this is what we do. This is how our research works. So we have an idea, and this gets written into a project where we say what we're going to do. And then we do what we say we're going to do. We get our results, and we publish both on a scientific level and for the general public. And then we take our results and we develop them into applications. Just so it's clear that we don't just get to develop the projects that we want to, but we have to submit our projects to competitions, and then we get external funding outside of our institute for doing the projects. My group has been fairly successful in the last few years, looking at national funding and European funding and Euregio funding. So that means we get results. Then we also collaborate with a number of parks and enti pubblici, so local governments, and that allows us to apply our results. And the institutional money we get is mainly used to develop, therefore, new protocols that we can apply to projects and so on. So most of the research money is still externally funded, even though we're not a university. And we are very lucky at the Fondazione MAC that for the last 10 years or so, we've had quite a lot of funding for our laboratories. So even though most of the group resides in this building, we can't keep all of our laboratories in the same building because a lot of our DNA is low quality and quantity. Our samples don't have a lot of DNA in them. So we have to avoid contaminating these samples by having different laboratories. So we have one laboratory for extracting DNA from modern samples, so tissue or soil, let's say. And then we have another laboratory where we extract DNA from fecal pellets or from feathers or from hair, okay, because there's not much DNA in that. And that's completely separated from this building. And then we have our little antique DNA lab way up here, where it's not anywhere near any other wild animal DNA. So we have a platform, but it's divided into these 3 laboratories. So that's how we work. Because we've been working on for 25 years on conservation genetics, we have databases and biobanks of a lot of different alpine species. This permits us to look at how different populations are isolated from one another, whether they have a good level of genetic variability, whether their genetic biodiversity is high, It also allows us to do wildlife forensics, because we can, if the magistrates come to us with a sample of deer that they think might have been poached, we can tell them which species it is and whether there's a probability that it's the same deer that the hunter had in his car last week. We can also, for example, when the couple of times that we had bear attacks in Trentino, we were brought the clothes of the person that was attacked, and from the saliva, we could tell which bear had been doing the attacks. So that's all based on the fact that we have gene banks for these species. We also have quite a few international collaborations. As I said, you can't do science isolated from everybody else. We have to collaborate with people from all over the world to compare our results, to learn things, to come up with new ideas together, as well as national collaborations, many in northern Italy, universities and so on. And then, of course, we have collaborations in Trentino Alto Adige, including with URAC and the university and the provinces as well. So as I've shown you now, my unit studies species biodiversity, genetic biodiversity, and now I will show you more about our work on microbiodiversity. So why am I studying microbiodiversity and conservation? How does that work? So first of all, what is the gut microbiota? The gut microbiota are all of the microorganisms that live in the gut. So we have bacteria, and we have viruses, and we have protists and archaea that all live together, thousands and thousands of species in our intestinal tract. For every one cell that we have on our body or in our body, we have 10 of bacteria and other microorganisms. So, and we have evolved with these microorganisms as mammals for 500 million years. So they are an intrinsic part of our biology. This microbiota can digest food for us. It produces vitamins. It helps the development of the digestive tract. It modulates the function of the central nervous system, and it stimulates the immune system. And it protects us against the colonization of parasites and pathogens. Having this colony in us actually stops other ones from being able to get in. So how does the host and the microbiota interact? It's really like we are our own ecosystem. We gain our microbiota from being born from another person with their own microbiota. We gain our first microbiota from our mother. And the passage to the outside in the world is extremely important for this. We gain microbiota mainly from our mother for the first days of our life. It's the same with wild animals. And then we start gaining microbiota from the environment. And our microbiota, in humans at least, develops for about the first 5 years, and then it's fairly stable. So our immune system, however, limits the growth of these microorganisms. At the same time, they stimulate the development of our immune system. So there's a feedback mechanism going on there continuously. But how do we study this? How do we study all of these species and complicated interactions? Well, the main technique now is called meta-taxonomics. And as I mentioned before for the metabarcoding, it's very similar. You take your DNA source, so in this case it's fecal pellets of hares, okay, which has the gut microbiota wrapped all around the outside and inside, okay. So this is where we want to get the DNA from. We want to know all of the microbiota that live in the gut of the wild animal, and fecal samples are a pretty good index of that. They contain most of the species that live in the gut. This has been shown. We extract the whole DNA from that, so all the DNA that's in the fecal pellets comes out. So in this DNA, there's a little bit of host, a little bit of diet, a few parasite DNAs, and a whole lot of bacterial DNA. So most of it is bacterial. We then focus on one part of the bacterial DNA called the V3, V4 in the 16S gene, okay? Details are not so important except that we focus on one little piece of DNA in all of these species. We cut that piece out and we sequence it in a machine that then tells us all of the species of bacteria that are in the original source. It's really quite amazing how this has developed over the last 10 years. The sequencing happens from this end, from the V3 end, and from the V4 end, and then using bioinformatics, so software, the two pieces are put together, and you get these little pieces that are about 250, 300 base pairs long that are read by a machine. And then there's a clever person who can do all the statistics, all the bioinformatics, and you need a big computer, okay? And then at the end, you get your list of microorganisms. You also get how abundant all these sequences are. So you can have both a list and how abundant all of these microorganisms are. And this is shown in a simple graph like this. So these are simple bar charts. Each bar represents one sample, and on the y-axis you have the relative abundance, so zeros. You can call this 100% if you like. And each of the colors represents one taxon, one microorganism type. In this case, it's just showing phyla. You can see Firmicutes, Bacteroidetes, etc. So you can see by the colors how many taxa are in here and how abundant they are. This is a very simple way of looking at it. But how could microbiota and how it works and how it functions, how could that affect conservation? Well, normally one thinks about extinction and survival of species in this way. When habitat fragmentation and degradation occurs or invasive species comes along, that's not good for species that are threatened. They can become small or isolated from each other, which means they breed together and get in more trouble. Their numbers could fall. This leads to even lower genetic variability and more inbreeding, more breeding together. They can no longer adapt. They might get a pathogen that kills them all and they go extinct. If we add problems with the microbiota in here, this is in theory, okay? If the biome depletion and gut dysbiosis, this means the biodiversity of the microorganisms is not high enough, and it becomes depleted and it doesn't function properly anymore, this could just add to the cycle of animals dying, becoming smaller, lower genetic variability, etc. So it just speeds up the extinction vortex. Okay, that is the theory. That is why we think microbiota is important to conservation. How much has this been studied? Hardly at all so far. Emily Pascoe, who was a PhD student with us, did a big literature review on this a few years ago. And she found that most microbiota studies focused on laboratory animals, many also on farmed animals, but only 5% were about wild animals or wild caught. Of course, that's increased slightly in the last 5 years, but not so much. Which taxa have been studied? And again, here we have the different taxa along the bottom and the number of studies. Most studies, 300, so more than half, have been studied on mammals and very few on birds, fish, etc., and hardly any on even rarer animals. And what have people studied so far? And again, most of the studies, this is a network analysis of the same literature search, which shows the different, all these big words are the different topics that they covered, and then the bigger circles are the abundance of publications on that topic. As you can see, most of them are about the composition, what's there, how diet changes gut microbiota, fairly obvious, how genetic background can affect it. In wild animals, again, there are mainly observations, so looking at how diet changes things, what's there, how different species have different microbiotas, fairly obvious. But in the meantime, the techniques were developed for wild animals, so now we can go on to some more interesting studies. Is there any proof that the microbiota actually improves the conservation of species? So does this diversity lead to any adaptive variation that could be selected by natural selection and help the species persist? Well, a little bit, okay? So far, in order for natural selection to work, we need variation in the populations. We need variation between individuals, as I explained before. No variation, no selection. Do we have variation in microbiota among individuals? Yes. Microbiota changes with age in chimps and in eels. It changes with season in buffalo and lemurs. We have evidence that there are specialized microbiota in different parts of the body, so the armpit or the mouth or the gut, they're specialized. They have different species. And we know that microbiota can change with diet. So animals that have different diets have different specialized microbiota. So there is variation. Other evidence that microbiota is adaptive is that we find microbiota diversity to be low where we expect it. So in captive bred animals, the diversity of the gut microbiota is very low, possibly because of inbreeding, possibly because they don't have contact with other individuals. So several captive animals have been shown to have low microbiota diversity. In natural populations, there's only one endangered species that's been shown to have naturally low microbiota levels. However, there are primate species, several now, that have been shown that where their habitat is disturbed, they have a lower microbiota diversity. And some other creatures where they have the microbiota at a low biodiversity on their skin, they are more susceptible to fungal pathogens. So there is some evidence that low diversity microbiota is not good. There is, in fact, some evidence that microbiota provides adaptive potential. So these are some examples of where microbiota is translated into phenotype, so something that can actually be seen and selected. For example, pathogen invasion is prevented in the golden frogs where they have a good microbiota diversity on their skin. The same with these turtles. They have a special microbiota on the outside of their eggs. And if the eggs don't have this microbiota, they don't hatch. Again, the bat example. If they don't have the right microbiota on their skin, they get pathogens. It has been shown that some phenotypes have to do with mate choice. So Drosophila will choose mates with a different microbiota. If you do a fecal transplant from a skinny mouse and put it in another an obese mouse, they will become thin. So this shows that there is some sort of phenotype being given to the host by their microbiota. Sorry. And as I said, the change in diet can lead to a change in diversity and loss of gut function. There are a couple of examples, only a few so far, that this change in diversity leads to the phenotype of a gut that does not work properly. In the pikas, which have a very specialized diet and specialized microbiota, and in these Zungwa red colobus that I will tell you about now. So there is some evidence that a dysfunctional microbiota or a functional diverse microbiota can lend adaptive potential to species, and this means that it could therefore have an effect on their conservation. Okay, so we've seen there is variation in microbiota composition. There's low variation where we expected if adaptive potential was compromised. We have shown that there are some phenotypes that can enhance survival or sustain gut function or change the phenotype, but for now there are no papers showing cause and effect of this. So there's still a lot of work to do in this regard. In my group, we have started working on this topic because I feel it's extremely interesting and neglected. We started with the project Ecobiome, looking at the interaction of worms and microbiota in a rodent model. This was a simple way to start so that we could develop the techniques that we needed to work on more threatened animals. So it was a sort of strategy to get the idea going in the lab. We then moved on to study wild primates in Tanzania. And then with Julia here, we got some funding to start studying the microbiota in ecosystems and how they interact. And just recently, we have moved this a further step to start looking at how microbiota changes through time when we're trying to turn the ecosystem back to something natural during rewilding. I won't be talking about that today because we're right at the beginning, but I'll show you a little bit about our results here. So, as I said, Ecobiome, led by Sarah Perkins from Cardiff University, was about how the microbiota and the macrobiota, or the microorganisms and the worms in the rodent model, were interacting. It hadn't been studied before, and Sarah is a parasitologist, so it was a good collaboration. And what we found was really quite intriguing. We looked at the microbiota in the different sections of the gut, so the stomach, the small intestine, the cecum, and the large intestine. We looked at all of those separately. And we also noted where the worms were living in the gut, the different worms of the rodent gut. And 99.9% of every rodent that you catch in the field has parasites in it of one type or the other. Almost all of them have the H. pylori, and quite a few of them have this one as well. So worms are very, very common. They used to be much more common in humans, of course, and there is some theories about how having worms at least once in your life protects you from some autoimmune diseases like type 1 diabetes and other autoimmune diseases. So the worm microbiota interactions are extremely interesting. So if you've had worms in your life, just consider yourself really lucky. If your kids get them, consider them really lucky. So, oh yeah, sorry, go back once. What we found here was that as expected, the sections of the gut, 3 sections, had very different microbiota. But what we didn't expect was that the presence of certain worms in these areas could change that microbiota. And even more unexpected was the fact that having one worm here was associated with changes in the gut of another part of the gut, both downstream from the worm and upstream. So having the Hymenelepis in your small intestine was associated with different microbiota in the stomach and different microbiota in the large intestine. So there was definitely something interesting going on here. Then we decided to look at some more yellow-necked mice a lot of worms and in their 4 different gut niches. And you'll remember these charts, right? Because we saw them at the beginning. The top row shows samples from stomachs, the bottom row from the small intestine. These are the names of the worms, and this is infected and uninfected stomach sections, okay? Infected, uninfected. You can, and each bar represents 1 worm. And the colors represent different bacterial taxa. So if you compare the 2 worms, they have a different microbiota. They're mainly green and blue, but they also have pink and yellow. So each worm itself has its own microbiota, which is somewhat different than the stomach itself. The same here. Worm, 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 infected and uninfected stomach. You can see the variety of microbiota of each of these worms compared to the stomach section itself. I find it quite extraordinary to think of these little tiny worms living and selecting their microbiota, what they need for themselves inside this habitat. So that was quite unexpected. Then we took these techniques that we managed to get working in our lab, and we started to apply them to more endangered species, which was the goal in the first place. So this Marie Curie project led by Claudia Barelli on the Udzungwa red colobus was the first one that we tried. She has been working out in the Udzungwa National Park for many years. There, she and her husband run a monitoring center out there. This is somewhat north, a little bit west. The Udzungwa Mountains are northwest of Dar es Salaam. So this is the pristine jungle that is only on these mountains, which in the middle of a lot of fields that have been cut. The jungle has been cut to grow sugarcane. So you have the intact forest, and you have this terribly disturbed forest where the animals are also hunted. Our hypothesis was that gut microbiota was essential for these monkeys' health, but that it depends on habitat quality, which is associated with different diets. And that if the habitat quality is compromised, we will have a reduction in gut function and diversity in the disturbed habitat. Let's see. So here we are, Tanzania, the Udzungwa Mountains, just a small area of these mountains left over from geological eruptions many years ago. We have the, if you can see this little square here, which is this one, so we have the intact forest, the national park here, all protected. We sampled from these 3 areas, so that's Manwana. And then we have this fragment, a forest fragment, where we sampled other groups of monkeys called Magumbera. So we have intact and fragmented. Manwana, as I said, is large, it's well protected, it's got a high tree richness. These monkeys eat only leaves. Magumbera is surrounded by human villages, cultivation, the monkeys are hunted, they're very stressed. When we go and look at their, oh, this is kind of funny because we didn't have to collect the samples. There are trained technicians in Tanzania who can go to a jungle like this and find these little pellets. They follow the groups of monkeys, the troops of monkeys, they stay way up high. They eat, and then they get to another tree in their home range, and they all poop at the same time. And then the technicians wait for them to move a little bit, then they run in and they look for these pellets on the ground. I don't know how they do it. I could see them if they're on the path, but not in the middle of the jungle. So I'll remind you that the forest fragment is the circles and the intact forest is the triangles. These graphs show the different groups of monkeys and then the number of different bacterial taxa in their gut. So from 300 to 500. This particular measure, the number of different taxa is called alpha diversity, exactly the same as when you study species diversity. Okay, it's alpha diversity. And as you can see, for 3 different measures of alpha diversity, the number of taxa, Chow-1 or Shannon, the forest fragment, the disturbed fragment, those monkeys all had a lower number of taxa than in the intact forest. So we do have a reduction of the number of taxa in the guts of red colobus living in the disturbed forest. But this particular paper went further. It was that this, look, don't read all this stuff. All I want you to notice is that these functions of the gut, which are given by the bacteria, this is just a list of functions, can be measured by what taxa are in the gut. So if you look at the functions that we were able to identify in the gut of monkeys from the disturbed forest compared to the intact forest, the functions, many functions were statistically higher in the protected forest. So not only did the monkeys in the forest fragment have fewer bacterial taxa in their gut, they had actually lost gut function. And the functions that they lost meant they were no longer able to digest some toxic plants. So the intact forest, the conclusions are the intact forest monkeys had a higher alpha diversity in their gut. The individuals living in the fragment lost some function of being able to digest these poisonous plants. So we concluded that habitat degradation does indeed impact gut microbiota variation. The next step here would be to show that this loss of gut microbiota function led to a lowering of the health and the fitness of these individuals. And does it mean that the monkeys in the fragment can no longer move back to the forest, or if they move back, would they just regain some function? This is really the holy grail of microbiota work, is actually showing that there's a, not only is there a difference, but this difference matters. And that's where we're going next in the future, because the technologies are moving ever farther ahead. We'll get there. Another study that I was involved in takes us now from Tanzania to Alaska, led by Sophie Watson, who was of Cardiff University. She was a master's student at this time. And she managed to persuade some Americans who go out every year and tranquilize and either radio collar or sample polar bears to look at various indices of their health, like their fat cover and their so on, to send her some fecal samples which they collected while they were examining these bears. And they sent us about 70 poop samples from the US, and she decided to analyze them to see if climate change was having an effect on their microbiota. Sorry, we have 112. So the hypothesis is this. These bears come from 2 places in Alaska. Because of climate warming, the ice does not, the summer ice is much farther out than it used to be. And some bears, instead of going out on the summer ice and eating seals and doing what they usually do, they stay on the land and they eat from bone piles, which are cetaceans that have been hunted legally by the native people, by the Aboriginal people, or they eat garbage, so they come close to human habitation. So you have what are called onshore bears, which eat this garbage, and offshore bears, which go and eat seals and their usual natural diet. So she wanted to compare the microbiota of onshore bears with offshore bears. And In this case, here you can see offshore bears, the ones that eat the seals, the onshore bears, the ones that eat the garbage. And again, this measure of alpha diversity, or an index of the number of taxa. And in this case, the onshore bears had statistically more taxa in their gut. And you can see that again from this chart here. The number of taxa in the onshore bears was 858. The offshore bears only had 635. They shared some of those. But the number of unique non-shared ones was higher in the onshore bears. So this was a bit perplexing because we thought he had it made with the monkeys. We said, okay, anything disturbed, they're going to have less lower diversity of microbiota. No, science is never that simple. The second example, we already have animals that are in a degraded habitat eating a different bad diet, and they have a higher microbiodiversity. So what we're looking at now is whether this higher microbiodiversity is due to actual good bacteria, or perhaps they have a lot more pathogens or perhaps they just have a lot more diversity because they come in contact with dogs, they come in contact with human waste, so they could gain more taxa in this way. In fact, interestingly, this same sort of result we see in the baboons in Tanzania, because they also go into the villages and eat human waste. So we get this similar phenomenon. What we do see, however, again, this is the alpha diversity, okay, Shannon. This is the total seal consumption, so the percent of seals in the diet, which you can actually associate with the amount of mercury that they're taking in, okay? The animals that eat more seals actually have, therefore, more mercury in their system and a lower microbiota diversity. So this could be something else that's going on. It's not that the garbage is worse than the seals, it's actually that the seals are actually feeding the mercury, and this is decreasing their microbiota. So it's still being studied. Sophie is still studying it, so we hope for a better explanation of what's going on there. And finally, I wanted to tell you about a bit about the project that I'm doing with Julia. And this is about the microbiodiversity of alpine pastures. So after studying single species, we decided that it would be really interesting to study the microbiota in a whole ecosystem. So looking at the microbiota of different creatures in an ecosystem and seeing how all of those interact. And this time, we were with the team. There's Julia. We had the team from URAC, from the University of Innsbruck, Paul Ilmer and his team, and us at the Fondazione MAC. And our sample site this time was a long-term ecological research site run by Julia and her colleagues above Glorenza, which many of you probably even work on and know about. So this was great because you have such long-term data on soil and soil organisms and meteorology. So we wanted to know if the microbiota in this pasture ecosystem changed with altitude, which we use as a proxy for climate change. So what's going to happen to the microbiota in pastures with climate change when it warms up? So we sampled at 1,000 meters, 1,500, 2,000, 2,500 at this site. And we wanted to know about this ecosystem level microbiodiversity, which hasn't been studied much, and define whether elevation affects the different indices of diversity. So again, we went out and sampled. And this time we had a proper experimental design where we had these 4 altitudes, we had 2 sampling seasons, we had 3 sites, so replicates on each elevation, and we had 5 samples for each per site. We sampled plants and their roots, So the rhizosphere, we sampled soil where there were no roots. We sampled the tiniest organisms, the collembolans and the nematodes in the soil, the earthworms and several beetles, which represented functional groups. And then we sampled some animals above the soil. There's no rodents here, it's too dry. So we sampled hares, which were everywhere, in the form of pellets, and then some domestic animals, sheep and cows, from their very large fecal pellets. And again, we went out in your beautiful province of Bolzano to these amazing places, and we spent some pleasant hours sampling soil and soil organisms and pellets. And just a few results to show you what we are finding, although we are in the middle of analyzing our 900 libraries right now, so these are just preliminary results. So again, this is alpha diversity, okay? So the number of taxa that we are finding in these different types of sample. Don't worry about these. This is just the names of the samples and the types. But look at this. This is the altitude, 1,000, 1,500, 2,000, 2,500 along the bottom. We have the soil and rhizosphere samples here, green and brown. We have the mammalian samples, so all fecal pellets, okay? Cows and deer. Sorry, deer are here, and these are horses. Cows, horses, deer, and hares. And then we have all the invertebrates over here. As you can see, we found that the soil and the rhizosphere had a far higher microbiota diversity than the other organisms. Mammals come in second, invertebrates are rather variable, but rather lower. But they're smaller, so that makes sense. Okay, so really high alpha diversity in the soil and rhizosphere soil. But this only tells you how many taxa there are. It doesn't tell you what they are. For that, we have to go to another diversity, and that's beta diversity. So taxa, an index that looks at taxa plus how abundant they are. And these kind of charts just show you, they represent the composition, okay? So what's in the sample. It's another representation of the composition. So in this project, we didn't only look at bacteria, but we also looked at fungi. Now, fungi, they're more difficult to study because they're not studied as much, so we can't name all of them yet. They're also difficult to study because it's more difficult to get the DNA out of them. However, they're an extremely important part of the microbiota in the gut because they don't stay there long, but they pass through and they're very stimulating for the immune system. So fungi and fungal diversity is really important for the immune system. So if you put all of the samples together, and the different shapes just represent different altitudes, okay, the soil and the rhizosphere composition is really quite different than all the rest. For bacteria. Fungi instead, the soil and the rhizosphere have a different composition to the cows. The wild animals have a different fungal composition, and the invertebrates are different again. So whereas they share an awful lot in the animals of bacteria, the fungi are much more specialized. The pattern is completely different, which was unexpected. If we go into more detail, this is a beautiful graph. So again, we're looking at beta diversity. We're looking at the composition of these different microbiotas in soil and in guts of animals. Again, we have the different, in this case, we have the different altitudes separated, okay? So we have, and this is all fungi, no bacteria here. We have 1,000 meters, 1,500, 2,000, 2,500 meters. As we go up the mountain, the soil and the rhizosphere have a different composition. This is very clear. As we go up the mountain, you can see the red ones, which are all the bovines. As we go up the mountain, they have a different composition in their gut microbiota. The wild animals and the invertebrates mainly do not change over the altitudes. So this tells us that there's an altitudinal change in composition, which is reflected in the bovines, but not in the other wild animals. Why could this be? Well, there is an association here with how far these microbiota can move. So in the soil in the rhizosphere, plants don't move, they sit there. So it's not perhaps so surprising that at each altitude they have their own composition. The bovines are stuck within fences up in Mallis. They're not free to move. So this is reflected in their microbiota at different levels. This is really interesting. Instead, the deer and the other animals, they can move, and so their microbiota is more uniform. The invertebrates are rather variable. Some of them have, if you enlarge this big blob of points, some of them you do see a little bit of altitudinal change. Some of them you don't at all. And which ones are those? The tiniest ones, the nematodes and the collembolans. So we think that those animals are being moved around with other animals a lot more than we already know. So what does this tell us about conservation of microbiodiversity and these ecosystems? So because we found that soil and plant and rhizosphere microbiota is very different at altitudes, we will expect climate effects on these microbiota communities. Will this have cascade effects on other animals and so on that live in the soil? Probably yes, because some of them, they share some of the taxa together. So if the soil changes, so will the microbiota of the other taxa. And we expect climate effects on the composition from alpine animals if their dispersal patterns change. So if the habitat is destroyed and they can't move around as much as they could before, their microbiota is likely to change too, because right now they can move everywhere. But if their habitat is degraded, if the climate means they can only live in certain places, they will probably have a changed microbiota as well. So the importance of corridors here takes on a completely different meaning. Corridors to allow animals to move between habitats. So that's just touching on some of the work that we're doing and trying to find out how important this microbiodiversity is for conservation. Some PhD students are now applying this to other species. So Francesca is looking at how chamois diet and microbiota and behavior are linked together. Lucia is looking at how skin microbiota has an effect on the amphibians in Trentino. Marina is carrying on with the work in Tanzania, and she is actually going a step further and studying metagenomics, that is sequencing all of the genes in the microbiota to look at function. Okay, so she's taking the next step, as I said. Lara is looking in the mountain hare, how microbiota and diet are related, and Chiara as well is looking at pathogens, diet, and microbiota and how they're related. So thank you very much for your attention. You've been very quiet. I'd like to thank all of our funding bodies, from the province of Trento to the European Union and Euregio, all the team at the Fondazione MAC who's contributed to this data, Claudia, the Cardiff team, and of course all the team at URAC and the University of Innsbruck. Thank you very much.